Hello. Anybody out there? I see one of you. Right. Nothing worse than having to look at your own face in these things. Okay. So good afternoon, everybody. We're going to be doing some Hardy Weinberg work today. I'll try to post up a video. I believe that there is some Hardy Weinberg stuff in Khan Academy that we can look up as well. I'm just going to pull that up on my other screen here. Just a second. So if you're having troubles with this section, there's definitely going to be some videos there. We're going to be referencing the worksheets that have been posted on the Google Classroom today. So hopefully you can get there and get to that classroom. I mean, if you're sitting at a computer right now, then hopefully you also can go to a classroom, get the files, print them off. If you need me to get them to you, well, I'll need to see you, I guess. So I know that some of you are in the building sometimes. We have classes till Friday here at the downtown campus. So feel free to come on down and access your classes there. So let me see what I've got here. I'm just opening up our file. So in the Bio40S classroom on Khan Academy, I'm looking, assignments. Let's see what it's, I can never really quite tell what I've assigned, what I haven't. So I'm not sure if I've done the evolution section yet, but I will pretty much just gonna click on this evolution section and add that. Assign everything. Great. All right, so I just assigned you the entire box of Khan Academy videos on evolution. We're not really gonna dwell into any of the beginning of life stuff. We just don't have time. It's really interesting, but they have a section on that as well. Let me know if you need me to talk louder or anything like that. If you want to tell me who's watching, I can't actually tell who's out there, and I can. Oh, look at that! Maybe I can. Nope. Okay, so if you want to drop your names in the chat or just say hi, that would be great, and then I can even mark you as present, so we can say that our class is that you're in class. So if that's something that's important to you, then go for it. All right, so today we're going to do some stuff on the Hardy Weinberg equilibrium, and I'm going to start by referencing this new document I just posted on Classroom. So it's from the Germany or the German Community College. I don't know how I came across this sheet, but it's a pretty solid one for this. And I like it very much for that. So this is a worksheet that says that has some nice little breakdown as to why we care about Hardy Weinberg equilibrium and some other ideas. So Hardy Weinberg equilibrium, also referred to as the Hardy Weinberg principle, is used to compare the allele frequencies in a given population over a period of time. A population of alleles must meet these 5 rules in order to be considered in equilibrium. So I often call these the conditions. Rules is fine too. Let's maybe just use my magnifying glass here. I like the word conditions. Conditions. So sometimes on a test or on an assessment, I've included things like name the 5 equilibrium conditions, or thanks for the update on your status of watching. I appreciate that. What if I might describe a population and ask which conditions of the Hardy Weinberg equilibrium are broken. So maybe we'll do some practice questions on that in a bit. So these are the 5 conditions. There are no gene mutations may occur. There must be no migration of the individuals either into or out of the population. Random mating must be occurring. So that means individuals mate by chance. There's no genetic drift. So there's the chance, there's no chance, a chance change in allelic frequency may not occur. May not occur. Not occur. And then there's no natural selection. A change in allele frequency due to the environment may not occur. It's weird that they got right on the first two. I don't know why they restated themselves. It's almost identical, but they've just sort of, you know, moved these. We should notice that these are the 5 agents of change that we assigned to our fingers the other day. So we have our gene mutations, which are middle finger, our migration, which are pointer finger. We have our random mating, which is the opposite of non-random mating, which was our ring finger, and genetic drift, which is due to small populations. And instead here, we're talking about large populations. So this usually implies that this is a property of large populations. So we could say that, I'm just going to make a little note here, because small populations I'm not going to get that in there, am I? Okay. Because small populations are more affected by genetic drift, equilibrium usually implies a large population. Okay, so I'm going to just put us all together into one sort of sentence here. Because small populations are more affected by genetic drift, equilibrium usually implies a large population. I'm going to get rid of that there. I suppose I could clean this all up after I'm done talking to you, but why? Nope, that was fine. Okay. So Hardy-Weinberg never occurs in nature because there's always at least one rule being violated. Hardy-Weinberg equilibrium is an ideal state that provides a baseline against which scientists measure gene evolution in a given population. The Hardy-Weinberg equation can be used for any population. It does not need to be unique. It's just a test that that can be used. If the population is in equilibrium or not in equilibrium, it doesn't matter. The test will still be used and tested for truth or not. All right, so there are 2 equations necessary to solve the equilibrium. Here, there's the first equation, P plus Q equals 1, and then the second equation, which is P squared plus 2PQ plus Q squared equals 1. So this is your math time. So I hope you all love math. I certainly love it. As I was talking about yesterday, these 2 pieces kind of describe different things. So this first equation, uh-oh. Okay, am I still up? It looked like I had an unstable connection there for a second. Okay, so the first equation describes the alleles. And then the second equation describes what we call genotypes. Predicted genotype. So ideally, if you were in a situation where you, yeah, you got a little bit of a notification that I was getting a bad connection there. Hopefully it's okay. It's stable enough. I think everybody in the building now is streaming every class, so hopefully we'll be okay. So genotype here is the combination of alleles. I'll try to speak a little slower, so throw it off, and I won't maybe make as many gestures. So genotype is the combination of alleles that an individual is carrying or a portion of the population is carrying. Meanwhile, the alleles are the forms of a gene that is being carried. So we have a lot of examples of dominant and recessive genes that we can talk about. Brown hair is often thrown around as a dominant allele. Brown hair versus light hair is usually a case of dominant and recessive genes. Dark eyes, all these sorts of things. So, you know, some good examples. What is it? Detached and attached earlobes is another example. So, you know, let's just go with dark eye color. And then the recessive allele is an example of a light eye color gene. Most genes that control the development of our bodies come in at least one or more forms. Very often they come in dominant and recessive forms. So for example, A-type blood is a dominant form of the gene that controls the form of carbohydrates on the outside of your red blood cells. And the O-type blood is due to a recessive gene that causes no carbohydrates to form on the outside of the blood cell. So there's another example of two alleles. They control the same trait. Alleles control the same trait, but they have different forms of that trait. Alleles control, or alleles can control the same trait, but with different outcomes. So for example, a population of cats can be either black or white. The black allele is completely dominant to the white allele. So we are going to identify them as the dominant allele and the recessive. So this is our dominant. And this is our recessive. Given a population of 1,000 cats, 840 black and 160 white cats, can we determine the allele frequency? The frequency of individuals per genotype. So to solve this problem, we're gonna solve for all of these variables, all 5 variables. So you got a lot of variables here, 5 variables, 2 equations, but really only 2 discrete variables, the P and the Q. So one thing we want to do is we want to first consider what we can do with these numbers. So we're going to make percentages out of these numbers. So we're going to take the total number of the individuals with your trait divided by the total population. So we have a frequency of white cats. We have 160 white cats divided by 1,000, or 16%. I'm a strong believer in allowing my students to represent their work in whatever format they like. If you like to work in percent, go for it. If you like to represent in fractions, do that too. So you could do 16 over 100 or 4 over 25, whatever you like. I think it might even be one more. So what I would like you to do is just feel free to use whatever representation you like. As long as I can understand it, then that representation is fine. And as long as it's accurate, I guess, is also the other problem. So once you know the frequency of the white cats, and most of the time for any of these problems, we're going to focus on the recessive gene. So we want to focus on the recessive gene. Focus first on recessive recessive genotype, since this is the easiest to count. As soon as you count up the number of cats that were white, you have counted up the white. You've got the white cats. You have their genotype. Just a second, someone at the door. So when we counted up 16 white cats, or 160 white cats out of that 1,000, we've got it. That's all of the cats, and we've actually counted up all of the recessive genes, or at least a good number of them. We just have to use a conversion to get that value out. So if we take the square root of q squared, because q squared is equivalent to the frequency of white cats, then by taking the square root of q squared, we can get the frequency of the recessive allele. Frequency of recessive allele. Now, the good news is that there aren't too many original types of problems in this example, in this section. So once you learn how to do it once, if you can successfully do it, do it once, you probably can do it every time. And so don't worry too much about, just focus on doing these questions a couple of times, and hopefully it'll come together. Now that you have the value for Q, you have essentially the frequency of the recessive allele. The frequency of the dominant allele must be the other part that adds up to 100%. Right? So if you've got a circle and you're telling me that 40% is Q, then of course it must be 60% that is P. And so that's what they're doing here. They're saying P plus Q gives you 1. So P must be equal to 1 minus Q. P equals 1 minus the 0.4 we just calculated. And that gives us P equals 0.6, or 60% dominant allele. Now that the allele frequencies in the population are known, we can solve the remaining frequency of individuals. We already know that Q is 0.16. So what is the remaining? The remaining has to add up to that difference. So the remaining must add up to 8.4. And so we see that if we take what it's written here, P squared, we take the square of 0.6, that gives us 0.36. And if we take 2 times 0.6 times 0.4, then we get 0.48. Now, this is also a little bit of a strange thing that we have not done a Punnett square yet, but certainly a Punnett square makes this make a little bit more sense. So I'm just going to draw one out in case you've done one before in a different course. But if you take a Punnett square, which is a tool used for showing the mating of two different individuals, and we put a P and a Q, and a P and a Q here, and treated them as multiplications. You may have even done this if you did pre-calculus, sometimes in multiplying two factors together. So thinking about this, that P plus Q equals 1, P plus Q equals 1, we're taking both of those equations and multiplying them. The result will be P times P in this box, which gives me P squared. P times Q gives me PQ. P times Q gives me PQ. And down here we get Q squared. There are 2 of these, so this gives us 2PQ, and that's the origin of this equation. P squared plus 2PQ plus Q squared is simply the probability of one mating partner having either P or Q, and the other mating partner having the same probabilities of P and Q, and multiplying them together, and we get a unique condition. This will not work for something like a Y trait, which is a trait that's carried on the Y chromosome, but we'll deal with that on another day. Actually, probably will never deal with it, to be honest. Okay, so let's add everything up together to make a statement. The frequency of the dominant allele, P, is equal to 0.6. The frequency of the recessive allele, represented by Q, is 0.4. This causes a frequency of individuals with the dominant genotype to be 0.36. The heterozygous individuals, 0.48. Recessive individuals at 0.16. So this is pretty interesting, right? This means, if we take this the implications here, is that you might have a trait that seems pretty small, right? If you hear somebody talking about something that's only found in 10% of the population, well, around 10% of the population, there are still about 50% of the population who are carriers. That's pretty wild. You know, if 10% of the population carried a disease, 10% of the population have a genetic disease, that means 50% of the population, or almost 50% of the population, are going to be carriers for that disease. So that's extremely significant if we wanted to study populations. So let's remember that frequencies can also be substituted back into the equations to double check. So if you think you're making mistakes, just take your numbers, add all of these ones together. They have to equal 1. Add both of these together. They must equal 1 as well. Okay, so let's do another question together. Oh, and then the final thing, of course, is that we were talking about cats. Right now, we don't even have a question about cats. So let's do some multiplication. We take the 16% that we found with the white cats, and we multiply it by 1,000. That's our original 160 white cats. We take the 48% times 1,000. That gives us 480 black cats. 36 times 1,000 gives us 360 black cats. So even though all of these cats were black, we know that about 480 of those cats are carriers. So comparing generations, to know if a Hardy-Weinberg equilibrium, to know if a population is in Hardy-Weinberg equilibrium, the scientists will observe at least 2 generations. If the allele frequencies are the same in both generations, then the population is in Hardy-Weinberg equilibrium. So recall, the previous generation had this, 0.6 and 0.4. That's exactly what we had just above. The next generation of cats has a total of 800 cats, 672 are black, and 128 are white. Is the population in Hardy-Weinberg equilibrium? So notice that we have a smaller population. There's only 800 cats now. So we're going to do some math. I'm going to stop for a second. Does anybody have any questions? They can drop them in the chat right now. Okay. I'm going to carry on a little bit. I just felt like I was going a little fast there. So I just wanted to make sure that you know that there's a chat. If you type in the chat like this, like we've seen already, then I can see that. Welcome to the other 4 of you who are there. Hope you're enjoying. So let's look at this example now. So we have 800 cats now. So we do the same deal. We're going to start. We can actually essentially use these as steps that are almost always going to be the same, especially if the question is set up this way. We're going to solve for Q squared. So we take the number of recessive genotypes divided by a total population, gives us 0.16. Use the idea that Q squared is equal to the square root of Q squared is equal to Q to solve for Q. So here's Q squared, taking the square root. This gives us Q. Once we have Q, we can compare this frequency. We can compare the recessive allele frequency, and we can see that it has remained the same. There's no reason to go any farther at this point because we already have all the evidence we need. All other numbers are derived from this Q value. So if we get the exact same Q, then they must be equal. So we can say they are equal. So no evolution. So no evolution has occurred, and we're still talking about fairly large populations. So it is in equilibrium. Okay, so let's do this next example. Let's see if we can figure it out. The beak color of finches has a complete dominant relationship with where black beaks are dominant to yellow. There are 210 individuals with the genotype of DD, which is 245, and the genotype of the heterozygous genotype of 245. Sorry, 210 is the homozygous dominant, and 45 is the recessive genotype. So remember that this is going to be our Q squared. This one is going to be P squared. And this value here is 2PQ. We need these values to be in frequency first. So this is our first step. We need a frequency. And frequencies are usually in decimal form or in percent form. So they added up, they found the population's 500, and then they divide each of the factors. So, or to find the frequency. So here, if we take individuals with the recessive population, we got 45 divided by 100, that gives us 0.009. We take the square root, that gives us 0.3. So notice that only 10% of the individuals are identifiable by their yellow beaks, but so this is like, we're saying 9% have yellow beaks. But when we compare that, when we bring that number through, 30% of the population, 30% of the pool is recessive allele. I don't know. I find that like a very fascinating observation. I still find myself trying to rectify that in my mind, that you have this obvious trait being carried by a small portion of the population, but out of the genes available, a large portion of them are carrying it, even though it's not being expressed. I think there's some interesting stuff to chew on there. I think you could think about that for a while, and you don't know. Yeah, I think you'd learn something. So use the first Hardy-Weinberg's equation to solve for P. So we take the difference, we get our P value. So this is our dominant allele. Now that we have the dominant allele frequency, we can solve for the dominant homozygous dominant. And we can even solve for the heterozygote. So that's our frequency for the homozygous dominant and our frequency for the heterozygous. So here we go. We can see that these two values add up to 100. These three values add up to 100 as well, or 1, I guess. And compare them. Now, this is a weird thing, is that they didn't actually go back and multiply them out to make sure they were right. Notice that we really only used one number. We just used the 45. We didn't even use the 210. We didn't use the 245. We just used the one single number. And if we look, though, yeah, I guess it would work. 0.49 times, what was it, 500? Yeah, I think so. Doesn't look quite like it's the right ratio, though. Yeah. Oh, no, I want times 500. Oh, okay, there it goes. All right, so it does work out. So they said 245 here, right? And so if we multiply 0.49, oh, they're back. Their numbers are backwards. Okay, well, anyways, we're going to ignore those. Let's just carry on with this example. The next generation of finches has a population of 400. There are 336 black beaks and 64 yellow beaks. So is the population in Hardy-Weinberg equilibrium. So this is the next generation, right? So we want to compare them to the next one. Okay, how did I get 0.3 from 0.09 in step 2? That is a great question. So this is always surprising, but this is a product of dealing with square roots, is that square roots of decimal places, the square root is always a larger number. So if we take the square root of 0.09, let me just point that up a little bit, then you'll find that the answer is 0.3. So if you had to do this by hand, we have to think that in the square root, we're finding the solution, or in taking 0.3 times 0.3, right? Which is the reverse of this. This is 0.3 squared, which is 0.3 squared. What happens mathematically is we have 3 over 10 times 3 over 10. So that's the same. These are the same values. And if you do 3 over 10 times 3 over 10, then we multiply the top, which gives us 9. And we multiply the bottom, which is 9 over 100. Well, 9 over 100 is the same as writing 0.09. 9 over 100. So mathematically, square roots of decimal values can be a little bit disconcerting, because usually when we square root something, we get a smaller number. But that's not the case in the case of decimal values. Anytime you take the square root of a decimal, you get a larger number. Does that answer your question, Caitlin? Excellent, thank you. All right, so then we have the same process. We'll see this happening here. We have the 0.16, and so we are going to end up taking a square root of that. And so I'm going to try to do it without rolling the page. I'm going to think about, okay, the square root. I'm doing the square root of 16 over 100. It's 0.6. And I do know the square root of 16 is 4, and the square root of 100 is 10. So that's 4 over 10. That means that it should be 0.4. And let's see. Yeah, that is the value that we get down here. So that's another way of doing it, is to take the square root of the fraction. And that sometimes makes it more obvious that why the value is going up. If you really think about what would it look like if we do the square root, what's the square root curve look like? Anyways, sure has a hump in it or something. Okay. Take the square root to find this, 0.4. Because the recessive allele frequency has changed, the population is not in a state of equilibrium. So we can see here, this 0.4 is different from our original 0.3. And the next steps for a biologist who knows this would be to study why. Why did it change? And that's where you'd have to get some ecologists involved, and you'd have to do a bunch of work and stuff. So yeah, so not change. This means that we could, we have some form of equilibrium, some form of evolution has occurred. It could be random. So probably biologists, the next steps would be to analyze this population again in the following year and see what happens. And if in the following year they notice a decrease in the same direction, maybe they notice that instead of 0.4, they get 0.42 or 0.45 or 0.5, and it slowly starts to move upwards, then they might suggest that it's a form of natural selection that's occurring. Perhaps this recessive allele is more useful at reproduction. Or maybe it was a random event, right? Maybe the next year they get 0.33. And then the year after that, they get 0.38. And the year after that, they get 0.46. And then when we start to add up multiple years and we start taking the average, we might find that on average, the Q is staying the same. And that's where multiple years of data are important for really getting into whether things are on a equilibrium or if they're actually changing over time. All right, so we are given some practice problems to try, and there are some answer keys at the end as well. So I think we've done some work here. I wonder if we could break now, and you could spend some time working on this this answer. In addition, on the classroom, I posted in the Hardy-Weinberg materials here, there is a second PDF here. If you could print that off and complete the answers and hand them in, that would be great. You can print it and take pictures of it. I believe you can even, I made it so you can edit on it. I'm not sure how that's going to work, but give it a shot. There are quite a few. If you feel like you get it after the first 3 or so, then just submit it, or just do a couple from throughout. You don't have to do the whole thing. You only have to do as many as you need to get the concept. This is a pretty long sheet. It's 12 questions. I would do some of the later ones to just sort of to show me in addition to some of the early ones, because those are a little tougher. But you know, maybe let's say, like, if I have to assign something, let's assign like the even numbers. Okay, try to do at least the even numbers. And if you want to do all of them, great. And if you want to do all of them and show me, even better. If you want to do just the even numbers, that will work as well. All right, I think I'm going to leave you to try those questions, and I'm going to give you the next sort of 20 minutes. If we were in class, this would be 20 minutes to work on this. So I think we're going to take that break now so that you can work on that. And I will be back tomorrow to go over the next thing, which is probably going to be talking about the history of the evolution theory. So the theory of why things happen. And possibly we'll post maybe we'll post, if you have any questions about this assignment, feel free to drop them in here, right? You can add a class comment, and you can just type in something. Like if you have a question about a specific problem, feel free to put in a class comment on the Google Classroom. And otherwise, let me know. If we need to, we can always set up a Google, what do we call it? A Google Hangout for the class. I might post that later, that we could create a Google Hangout where you could drop questions more regularly, and we can even have little meetings in there as well. That would be more interactive. But for now, we're going to stick with these live chats so that the video stream is saved and it's available for other students. Remember, this will be going on for all of next week as well until we hit spring break. So will be the plan. So probably trying to aim for half an hour to, I'll really try to avoid doing hour-long lectures online. I don't think anybody wants to sit at their computer for an hour. So we'll try to aim for about a half an hour lecture with some sort of assignment that you can work on for the other half an hour. And that'll be kind of the plan going forward until we are back in regular school hours. Okay, best of luck out there, and we'll talk to you soon. Bye.